Hey y'all, welcome to part 3 of your lecture series on methods. We're going to talk a very little bit on experimental design. I just kind of want to go over the big stuff, the big points. So last time, previously on the method series, we talked about the limitations of correlations, and we talked about the fact that if you really want to get at causation and not just correlation, you need an experimental design. So, some of the most important things in experimental design are variables, right? There's an independent variable, you set it, you control it. You're also going to have a dependent variable. Dependent variable, you measure what happens to it. Okay, so here's a good example. Developers, when they're trying to design a website, they'll often design 2 different versions of the website and give it to 2 different groups of people, and they'll see, so for instance in this particular experiment, you want to see if I put the buy now button here, do I get more clicks than if I put it here? I want people to click on this buy now button. So I give each of these to a separate group of people and I compare how many clicks I get between the groups. This is called A/B testing. Developers use it all the time in human factors, but really in science it's just called experimentation, right? You have a variable that you change. In this case, the independent variable is the location of the buy now button, and you have a variable that you measure, and in this case it's the number of clicks that you get based on where the button is. So the next important component of experimental design is your sample. So when you choose a sample, you're going to have 2 different options, really. You're going to have a case study. This is where it's just one person, it's a single individual. This is good for some things, right? Rare or familiar, like unfamiliar disorders, things we've never seen before. So if this person has some kind of weird disease that we've never seen, we want to record the progression of the disease and take some data from this one person, so at least we have some initial observations, right? That's the benefit of a case study. But, ladies and gentlemen, when you can get more than one data point, please do it. Okay, at least most of the time. I can't really think of a situation off the top of my head where having less data is better than having more. Okay, so you're going to want to, when you can, choose a random sample. Now, a random sample is where every member of a population has an equal chance of being included in the sample. So what's the difference between a population and a sample? A population is sort of the collection of people, of participants that you might possibly recruit from. So when we run studies at UT Dallas, we recruit from the undergraduate student body, right? This is the population that we're picking from. Those of you who sign up for a particular study, that group of you in that study, that's a sample. You are part of the sample. It's very important for generalization that the population you're sampling from is representative sort of the world, right? Like we are the world. We want this in our sample because we want to be able to take our data and say, look, we tested this on a population with a variety of genders, a variety of physical appearance and social status and socioeconomic status and grade point average and all these kinds of different variations in these people so that we could represent a lot of the population. Now, there is a trick there because if there's a consistent variability in your population, that can lead to skewing your data like behind the scenes, right? It's going to mess with your data behind the scenes. So we always check all the variables about our population to make sure that, or about our sample, to make sure that none of these variables, demographic variables, what we call them, are going to be affecting the data. Okay. So, important for generalization, right? Finally, well, maybe not finally, I think there's one more point after this, but okay, when you have your subjects, your sample, I'll get there. Okay, so once you have your sample, you're going to want to divide them into a control group and an experimental group most of the time, okay? So here's my sample. I've got a bunch of little chubby leg kids here. and I divide them randomly. I just sort of randomly pick one person to be here, randomly pick one person to be here. Okay, this is my experimental group and my control group. And what I'm going to be varying between these two groups is the independent variable, right? So my independent variable is whether or not I expose these groups to media violence. So my experimental group gets exposed to media violence, and my control group does not get exposed to media violence, okay? So my results are high aggression for my experimental group and low aggression for my control group. Now, let's say I want to test mediating factors, right? Mediating variables. So I have my sample, okay, and I divide my sample randomly into two groups. But now what I do is this. I assign half of my experimental group to one subgroup, right? And the other half to another, and the same with my control group. Now this subgroup is going to be based on adult supervision. So half of each group gets adult supervision and the other half does not. So we look at the effects that adult supervision has on the outcome, and we see, at least for this fictional table of results, that adult supervision as a moderating variable still leads to high aggression, and we still get low aggression in our control group, right? So this is a stronger design than the previous one if we're looking at causation versus correlation. And finally, it's the mother of all components when we're talking about experimental design. It's significance, and this is the goal. This is the goal of every study, is to be significant. So we do all these fancy formal statistical analyses, and we make all these assumptions about the data, and we make sure that the data is equitable on both sides when we're looking at controls versus experimental groups, right? But really what we're trying to get down to, we just want that one number that tells us that our numbers are significant. And significance really means not how good it is, but what are the odds that it's not good? What are the odds that our experiment sucked? It just failed, right? So when you have really low odds, when the p-value, the probability that your methods failed, is really low, that's really good, okay? That means your results are significant. Any effect that you're measuring there is a significant one. Now, that's not to say that you have to achieve statistical significance in order to be important. I'm sure you can come up with examples for your interaction, maybe, about results where finding no difference between a control and experimental group would actually be something worth noting, right? So if you think about something like that, and you come up with an experiment in which that might be a good thing, you could use it as an interaction. Okay, so that was a super short little lecture. We're going to move on now to actual technical measures. So we're going to talk about things like how are we actually measuring what's happening in the brain to make assumptions about the mind and cognition. We'll talk about neuroimaging and et al., right? Okay, so I'll see you guys in the next lecture.